Alzheimer's disease, the most common form of dementia, is characterized by progressive generalized deterioration of the brain. The hippocampus, a region of the brain responsible for the consolidation of information, spatial navigation, and the formation of short and long-term memory, is affected early in the course of the disease. The two most prominent theories about the mechanisms of Alzheimer's disease include the accumulation of extracellular beta-amyloid aggregates and the intracellular deposition of tau protein tangles in the brain. In a healthy brain, amyloid precursor proteins, or APPs, on the surface of a neuron are cleaved by an enzyme called alpha secretase, releasing an APP fragment. Another enzyme called gamma secretase cleaves APP again to form a second short fragment. The function of APP and these protein fragments is poorly understood. In Alzheimer's disease, the amyloid precursor protein is cleaved by an enzyme called beta secretase at a different location. When APP is subsequently cleaved by gamma secretase in Alzheimer's disease, the result is a longer second fragment called beta amyloid. These longer beta amyloid fragments aggregate into insoluble oligomers, which then accumulate to form beta amyloid plaques in the surrounding neural tissue. This accumulation results in oxidative and inflammatory injury and ultimately synaptic failure. The buildup of beta amyloid plaques may be the initiating factor in Alzheimer's disease. The role of tau protein in the second proposed mechanism involves damage to neuronal microtubules. Inside healthy neurons, microtubules provide structural support and act as a platform for intracellular transport. Microtubules are composed of tubulin and stabilized by tau, a normally soluble protein in the axonal region of the cell. In Alzheimer's disease, tau proteins are hyperphosphorylated, a process that makes them insoluble and causes them to lose their affinity for microtubules. This process destabilizes the microtubule structure. The abnormal tau proteins self-associate into paired helical molecules, which may be cytotoxic. These abnormal molecules aggregate into insoluble filaments that form neurofibrillary tangles. The tangles accumulate within the cell and are a marker of the severity of Alzheimer's disease. The dissociation of the tau proteins and the destabilization of the microtubules disrupts the transport system of the affected neuron and may lead to cellular injury and eventually cell death and synaptic loss. This pathologic process causes inflammation and oxidative damage, resulting in atrophy of the hippocampus and cortical regions of the brain, as well as hypertrophy of the ventricles. Clinical manifestations of Alzheimer's disease may include impairments to memory, language, executive functions, behavior, and visuospatial skills. The underlying causative mechanisms may be diverse. No single linear chain of events has yet been traced in the onset and progression of this disease. The understanding of the pathophysiology of Alzheimer's disease remains incomplete.